Welcome to Biochem Plus. In the next 2 minutes, you'll understand the essence of glycolysis. What is glycolysis? Glycolysis is the pathway that breaks one glucose molecule into 2 pyruvate molecules, producing 2 ATP and 2 NADH. It happens in the cytosol, and it does not require oxygen. That means every cell can use glycolysis, even in low oxygen conditions. Why it matters. Glycolysis provides quick ATP for immediate energy, pyruvate to fuel the mitochondria, intermediates for building amino acids, lipids, and nucleotides, the only energy supply for red blood cells. It's essential for the brain, muscles during exercise, and rapidly dividing cells. How glycolysis works. In simple steps, glycolysis has 10 steps, divided into 2 phases. Phase 1, energy investment. The cell spends 2 ATP to modify glucose and split it into 2 identical 3-carbon molecules. Phase 2, energy payoff. Each 3-carbon molecule generates ATP and NADH. In total, the pathway yields 4 ATP, so the net gain is 2 ATP per glucose. Finally, the pathway ends with the production of pyruvate. What happens to pyruvate? With oxygen, pyruvate enters the mitochondria and forms acetyl-CoA for high-yield ATP production. Without oxygen, it becomes lactate, allowing glycolysis to continue. 3 enzymes control glycolysis. Hexokinase, PFK1, the major regulator. Pyruvate kinase. They respond to ATP, AMP, citrate, and hormonal signals, ensuring glycolysis speeds up when energy is low and slows down when energy is high. Thank you for listening. Do well to like, share, and subscribe to this channel for more simplified pathways.